Thanks, Rue. What I want to do is focus on science question 1, which is how do upper ocean ecosystem characteristics determine the vertical transfer of organic matter from the surface ocean. And by ecosystem characteristics, we're including community composition and also the physical environment. And I'm going to focus on question 1 in the upper ocean, and Adrian Martin is going to focus on the twilight zone. So there are, in addition to the main question, there are 4 sub-questions as part of question 1. How does the plankton community structure regulate export from the surface ocean? How do the 5 pathways? Whenever I talk about the pathways, I always feel like I'm doing a Dale Carnegie speech, that this will get you to heaven, but this will get your carbon out of the upper ocean. So what are those 5 pathways that drive export, and how do they vary with the community structure? What controls aggregation and disaggregation of particles, and how are these controlled, influenced by the plankton community structure? And you notice there's this repeat in terms of plankton community structure. And the last sub-question is, how do the ecological, physical, and ecological processes act to export organic matter? Here's a blow-up of the wiring diagram, just focusing on the upper ocean. And as Ruth said, there are 3 sinking pathways: sinking of intact cells, sinking of aggregates, sinking of zooplankton byproducts, vertical submesoscale advection of DOC and POC, and then transport of organics via vertically migrating organisms. So those are the 5 pathways. And what I want to do in this really short talk, and I've emphasized them in blue, is talk about a couple little vignettes in terms of what have we learned in the last year, and what are some of the challenges, and how could this fit into the larger exports picture. And what I'd like to do is invite you to think about the rest of the talk as a walk through a sculpture garden, where you have a 2-year-old in your hand, and you're trying to get the big picture, and you're not trying to dissect a Rodin. So I'm not going to go into all the details on these slides, but what I'd like to do is convey for 3 subtopics: plankton community structure, the taxon diversity, and the submesoscale patchiness. Carbon export depends on community structure and submesoscale physics, and the DOC-POC continuum is influenced by community structure and export biophysics. And again, coming back to this idea of community structure and the submesoscale physics. The first sculpture is a recent paper by Levy and colleagues who looked at, using the Darwin model, how the plankton diversity is influenced on the basin and the mesoscale. And what I stole from their paper is that small scale diversity, and they're thinking about what happens with fronts and eddies, is that it's the large scale conditions set the broad biogeochemistry. Horizontal stirring dynamically distorts the large scale niches, and biological and ecological responses on the time scale of fronts and eddies are controlling this diversity. Now, one of the points that they made in this paper is that there are relatively few real data sets that allow you to examine that. And there are 3 papers that were published either this year, actually next month, and then last month and last year, that have different aspects of looking at community structure that come from the North Atlantic bloom cruise. And one of the points of the Mahadevin paper is the great patchiness that you see in the subpolar North Atlantic, and in part based on the mechanism for setup of the bloom. So the first one was done with old-fashioned phytoplankton cell counts with Udimol settling chambers. And these were samples that were collected daily over the course of a month. And the conclusion of this paper by Mosing et al. That's coming out next month in Journal of Ecology is that the phytoplankton community structure was set by the subscale, mesoscale frontal dynamics. And that controls alpha, the local diversity, and beta, the temporal diversity. And the subscale physics, the differences in salinity between the high and low salinity regions are less than 0.08 parts per thousand. So we're talking about subtle but important differences. And what they found was that 31 taxa were shared in both water masses, but 3 were found only in the low salinity water and 22 in the high salinity water, and that this structure was maintained by the sub-mesoscale physics. Now, taking a very modern approach, Bestward and Van Oostenen, if I'm pronouncing that right, used something on the order of 250 DNA primers to look at functional genomes for phytoplankton related to nitrogen assimilation and carbon assimilation. And he had these phytoplankton arrays, and again, it was taken from very similar times in the North Atlantic spring bloom, what they found, and so this is a really well-studied phenomena, but they found that a few archetypes dominated, showing very distinct temperature and salinity patterns from other archetypes, suggesting that a few species dominate against a high background of diversity. Now, one of the challenges that I think we have in terms of how do you get at community structure is how do you merge the Udermol, who the species are, with what their functions are. Yet another way to look at diversity is the big challenge of how do you scale up? You go out to sea and you measure a few highly detailed measurements of community structure, but they're really expensive. So how do you scale it to the larger spatial scale? And one of the things that Sedanick did was looked at the ratio of optics, chlorophyll fluorescence to backscatter, and found that there were 2 different communities that related to the very detailed measurements that Mike Saraki and Nicole Poulton did on the shipboard, and was able to say a high ratio related to diatoms and a low ratio related to non-diatoms. And what they did was develop a heterogeneity index that showed before the bloom there wasn't very much heterogeneity. So it's mapping shipboard measurements onto the larger scale using gliders and simple optics on gliders, and showed a great diversity during the bloom, and then after the bloom that diversity crashed. And I want to come back to that because that's also a way that one can help to scale up measurements of export flux. Megastapa is going to talk about the sampling array, and one of the key aspects of exports that makes it different, that advances the science, is the fact that it uses a diversity of scales. So it's a nested sampling using ships, gliders, floats, and satellites. Second vignette, as our walk through the garden, is that export depends on community structure and submesoscale physics. A paper just published by Agudi from the Tara expedition shows an estimate of flux that they used from taking particle size estimates with sinking particles. And you can see the continents and the cruise track. And so they came up with these large scale global estimates of carbon flux. And then they asked the question, because they took all these genomic samples, is what is the community? Not just saying, oh, it's diatoms or it's recyclers or whatever. They did very sophisticated network analysis to ask what kind of community dominates high flux or low flux. And it's a complicated paper, and I think it's just the beginning of a new way of doing things. But what it shows is that the importance of community, not just of particle size in flux. And this paper also opens up a whole new area of questions for viruses and for small particles. In the Southern Ocean, there are inverse relationships between NPP and export. And what Lemoyne et al. Did in a recent paper is to look at sort of pre-averaging in terms of the amount of surface recycling and grazing, where they had low surface recycling and high grazing, low surface recycling and low grazing, and high surface recycling and low grazing. And what they suggested is really trying to think about the community. You know, it takes a village to raise a child. Well, it takes a community to flex some carbon. And, but one of the things that they were not able to address is what's the role of DOC quality in terms of that recycling. Again, going back to the Mahadevan paper where we see the patchiness, what was shown in this paper is that the mechanism for the start of the bloom inherently leads to a patchy bloom. And so how then do you wind up scaling up? This is a really busy picture. The take-home points are that when we're using Lagrangian float and measuring net community production and the difference between the accumulation of carbon and the accumulation, the projected accumulation from NCP, is that in a dominated diatom community, export flux was high. In a recycling-based community, export flux was low. Using the optical index from the Sednick paper, what Eric DeSaro was able to do is apply these export rates based on the community structure. Now, one of the limitations of that particular study was that the sampling schema was not really designed to come up with spatial mapping, but that is possible with exports based on the sampling array. Another example is a recent paper by Megastapa, who showed the relationship between net community production and export flux based on point-to-point measurements. Point measurement of one, point measurement of the other. And Adrian can tell me if this is the right application that I've heard people from the UK call it. The data set looks like a spotted dick, which I think is a pudding with raisins, and that's what I've been told. But if you average over the transects, a very nice relationship between export production and NCP emerges from that. So the sub-mesoscale physics is really important. And one of the things that exports does to advance science is this nested array that really addresses the physical environment. And lastly, what I'd like to do is talk very briefly on how the DOC-POC continuum is influenced by community structure. This afternoon, Monica Oriana is giving a talk on the DOC-POC continuum. And DOC is an operational term. I call it a garbage can term because it basically it's operationally defined by what the filter size is. But in reality, DOC is individual DOC polymers that can become annealed into microgels, that can become annealed into larger gels. So it's sort of like a tangled network. So it's a different way of thinking about DOC and POC in this continuum, and the size makes a difference in terms of its accessibility. One of the things that Monica found in the Southern Ocean is that a diatom-dominated community produced big gels, and a prokaryotic-dominated community produced little gels. So the quality in terms of the physical size makes a difference, but also the quality in terms of what's produced. Now, this is a laboratory study by Weir et al., where they showed that silica-limited diatoms produced DOC that was rapidly metabolized by the bacterial community. And so silica versus nitrogen limitation has a difference in terms of the amount of DOC produced, but it was very labile. But then what they also found is that when it came to longer-term persistence of DOC, it was the species that mattered, not the type of limitation. And if it has a longer-term persistence, then it becomes more susceptible to physical export. And the last slide that I want to show is thinking about how DOC and particles can be, on the mesoscale, subducted down into the twilight zone. This was a paper by Oman last year where she had a combination of a model and glider observations, again from the North Atlantic, that showed that there were blips of water that were carried down at the edge of submesoscale eddies to 200 and 300 meters that preserve the DOC and the POC content of the surface water. And I know Adrian is going to touch on this, but one of the big questions is, we know something about the spatial patchiness of phytoplankton because we can see them with ocean color. But what we don't know is what the spatial patchiness is of microbial communities in the deep sea, because it's very difficult to assess it. And so maybe there are hotspots that are based on these subduction mechanisms that carry parcels of water down. And so I'm going to turn it over to Adrian. And again, I hope you enjoyed your walk through the garden.